We already heard from Darwin, but next we're going to be hearing from Dr. Anthony Barley, who is an assistant professor in the School of Mathematical and Natural Sciences at ASU. Dr. Barley grew up in California and he received his Bachelor of Science degree in Biological Science from California State University in Sacramento. He also received a PhD in ecology and evolutionary biology from the University of Kansas in 2014. And after graduating, he went on to do postdoctoral research at the University of Hawaii, where he was awarded a postdoctoral fellowship from the Arnold and Mabel Beckman Foundation. He also worked as an assistant project scientist at the University of California, Davis, before joining the faculty at ASU in 2022. Dr. Barley studies the evolution and conservation of reptiles and amphibians in the southwestern United States, southeastern Asia, and Mexico. He's going to be discussing how digital mapping, iNaturalist, and genomic tools are transforming herpetology research.
Hi, everyone. Thanks for having me. My name's Anthony Barley. I'm an assistant professor at Arizona State University. Today, Alyssa invited me to talk a little bit about how technological advances are transforming herpetology research, in particular, the research that I do as a scientist. I'm going to talk about three different areas today. I'm going to talk about this in the context of digital mapping, about new genomic technologies, and then I'll finish by talking a little bit about my teaching at ASU and how that's been affected by some technological advances. So, one of the major motivating factors that underlies all the research I do as a scientist has been a fascination with the remarkable diversity of reptile and amphibian species that we share the planet with. So there's just a few pictures of me throughout my career. You can see back when I was younger, I had some less ideal style choices, but the picture with me at the giant steak was my first time I ever left the country when I went down to look for reptiles and amphibians in Mexico. The top left is a picture of me on my first trip to the Philippines as a graduate student, where I found Philippine flying lizards. So it was just a mind-blowing experience walking through the rainforest and seeing lizards gliding over your head from tree to tree. On the bottom left is a Mexican mole lizard, which is a species of lizard that only has front limbs. It lives underground. It's fossorial. Every day when I go outside and find horned lizards here in Arizona, where I'm a professor, I'm still blown away by them. This is a knob scale lizard, me and my first knob scale lizard. These are weird, wacky lizards that live in rock crevices, in outcrops in Central America. In Mexico, they have really low body temperatures. They live a very long time. They're super interesting. This is me looking like a crazy person when I found my first Bell's salamanders. These are, again, giant salamanders from Mexico that are jet black and they have these really pretty red chevrons on the back. And then this is a crazy looking Mexican burrowing toad. So these are just a few of the incredible species that I feel like I've been lucky enough to encounter during my time as a biologist. Um, but in addition to the truly remarkable and interesting species that we know about on planet Earth, it turns out that there's a huge amount of diversity that is still unknown to scientists. So, although we know a little bit more about reptiles and amphibians, if we think in the context of all species, probably only 10% of all species on Earth that exist have been documented by scientists. Much of that diversity is distributed in tropical areas, including in what we call biodiversity hotspots. So these regions of the world in red here, where a huge amount of species diversity exists. And so I'm going to share with you a couple of my field experiences from my work, where I've mostly worked in the Philippines, here in Southeast Asia, where we have one star, and then in Central America as well, both of which are biodiversity hotspots, where we're still learning a lot about new species of reptile and amphibian there. So, finding and describing new species of reptile and amphibian has been a major part of my career ever since I started as a graduate student. So on the top left here is the Philippine orange-spotted stream frog, a new species we described based on my first trip to the Philippines, where we were hiking in the mountains high up in the northern Philippines, and we came across this strikingly beautiful bright green frog with orange dots. You can see here. Here's the Isan fang frog, another species we described, which is a frog that actually has these large fang keratinous teeth in the front of his jaw that it uses to find prey and eat prey. And then the most of my work as a graduate student at the University of Kansas focused on learning about these sun skinks, which you can see pictured here. Here's four different new species of sun skink we found and described from the Philippines. Over the past decade or so, I've spent more time working in Central and South America. So here is a new species of knob-scaled lizard, one of those crazy looking rock crevice lizards I mentioned that we described. This is the Aguaruna marsupial frog from Peru, a species of frog that in which the eggs develop in a pouch on the back of the female, just like a marsupial. It's a species of frog, though. And then most of my work in the recent years has focused on studying whiptail lizards. So here's four different whiptail lizard species that we've described in recent years. So a lot of incredible diversity of reptiles and amphibians that we're still learning about on planet Earth. Um, Documenting and describing new reptile species requires first that we learn about the discoveries of previous scientists, since I'm not the first person to work in many of these areas. So, like I mentioned, my graduate research was focused in the Philippines, which is an island nation in Southeast Asia. You can see it here on the map. It's an incredible area that has a lot of different types of habitats with rainforests and cloud forests and active volcanoes. But it also has a really interesting history of research in herpetology. So, the first thing you learn about when you learn about the reptiles and amphibians of the Philippines was that one of the first biologists to study them was Edward Taylor, who's a really prolific herpetologist. He described over 150 species of reptiles and amphibians during his career. But he was also did some really interesting work as a covert CIA agent. So at the time during World War II, when he was actively working as a herpetologist, he was contracted by the CIA to gather intelligence information from different parts of Asia and Siberia, and even was responsible for training CIA agents in jungle survival in Sri Lanka. So, super interesting character who's also a really interesting herpetologist. And so when I went to the Philippines to start studying the sun skinks, some of these species had not been seen by scientists since Edward Taylor described them. And historically, how we've gone about learning about what previous scientists have done is focused primarily on reading the papers they published or the books they wrote. But now that we have digital platforms like iNaturalist, where citizen scientists from anywhere in the world can post their observations, we actually have opportunities to plan our fieldwork around those observations that non-scientists who live in these areas have posted. So here's a couple species of sun skinks that you can find on iNaturalist that biologists posted that before I started working there and I went to go to these areas to collect and sample tissues of these species. Biologists hadn't seen them since Edward Taylor had seen them, but many of these areas have changed in 150 years. So this provides an opportunity for us to see, do these species still exist in the area we want to go visit and sample them? And it provides a really great opportunity for us to plan our fieldwork better. These are all the iNaturalist records from Mexico and Central America. You can see in red the iNaturalist records of whiptail lizards, I should say. And so on the left here is a picture of a whiptail lizard that I found an iNaturalist observation of that had never been before documented by scientists. So this is one of my, what I think of as the prettiest whiptail lizard that I've ever seen. You can see it's got these beautiful bright colors, but it's also got like a leopard print spotted pattern. Um, Beautiful species. So I was looking at the records that locals had found in Mexico on iNaturalist and came across this, and then planned some field work to go sample this species since it had not been documented by scientists before. This is the Mexican pygmy whiptail lizard that we went and sampled a couple years ago. This is a species that was described in the 60s, only occurs in one small valley in southern Mexico, that is heavily kind of changed by agriculture over the past several decades. And so, again, it was an opportunity for us to look on iNaturalist, see, can we find these species in these areas, or have their habitats changed too much? And allows us to really plan field work, which is really great because this is really logistically complicated work to visit many of these places and sample their lizards. Another way in which I've been using iNaturalist data is to identify new species introductions. So I'm originally from California, so I have a particular fondness for the species there. But over the past few years, these are a couple iNaturalist observations that were posted. One's from Northern California and one's from Southern California. So California only has really two native species of whiptail lizards, neither of which is these. And so I came across these pictures, got in touch with citizen scientists about them, said, hey, you posted this observation of this whiptail lizard, but that kind of whiptail lizard actually doesn't live in California. And so we went there and worked with the citizen scientists and sampled the species, and we're currently working on using genomic data to try and understand where did these lizards get introduced from, since they're actually native to different parts of the southwestern US and northern Mexico. All right, so that's a little bit about how technological advances are transforming herpetology research, in particular the context of how we can use digital mapping to revisit historical localities, find new populations of species, and track species introductions. So next I'll talk a little bit about new genomic technologies. Um, so one of the really cool things that really rapid advances in genome sequencing over the past decade has allowed us to do is that we now can read thousands of DNA positions in the genome of virtually any animal at one time. So you can think about the process of DNA sequencing as analogous to putting together a puzzle. What we can do is read long, different segments of the genome, and then we need to try and figure out how those different pieces of the genome fit back together to understand how to use them. And essentially, over the past decade, what genomic sequencing technologies have allowed us to do is go from trying to put together a puzzle that is millions of pieces to putting together a puzzle that's only thousands of pieces. So if you think about putting together those two different types of puzzles, it's a lot easier to think about putting together a 1,000-piece puzzle than a million-piece puzzle, right? And so this is really cool because it allows us to study the genomes of almost any animal on Earth. And so now we can go out and so these are some pictures of me in the field over the past decade or so working with a bunch of biologists. So all of the research that I do is in collaboration with scientists from different countries. So these are pictures of me and scientists who have helped us out in Mexico, as well as students. Um, and these are some pictures of some whiptail lizards that we've gone out and collected. So yeah, I should mention that all of this work is based on working with undergraduate and graduate students in different countries, and that's a really exciting, important part of my work. But now that we can sequence virtually the genome of any animal in the world, it means that we can uncover many of the secrets about their lives that we didn't know before. And so, as an example of this, one of the things that's always fascinated me is that among the about 60,000 species of animal on Earth that have a backbone, although most of them have males and females, there's actually about 50 or so species. These include fish, frogs, salamanders, and lizards, and even a snake, of which only female individuals exist. And these females are really remarkable because they reproduce themselves by laying eggs that don't need to be fertilized. So they lay eggs, and those eggs develop directly into fully functional adult lizards. And those lizards are clones of their mother. They're identical to their mother. So it's a really fascinating biological phenomenon. The largest group of those animals is the whiptail lizards that I've been studying that I mentioned. Here's a picture of one on the top left. So, using DNA sequencing technologies, what we've been doing is reconstructing the entirety of the family tree of these whiptail lizards. So, I won't show you all of them. This is basically what the family tree of whiptail lizards look like. There's about 50 or so species of these lizards. And this is really cool because it is a question that biologists have literally been trying to figure out for about 10, or excuse me, for over 100 years now. And we've only recently been able to resolve this question of how the different species are related to each other due to these advances in genomic technologies. And so here are 10 of those species of whiptail lizards. These are the really unique special ones I mentioned. These are the entirely female species. There are no males that reproduce by laying eggs that develop into clones of their mother. So they're remarkable species, and by having this information, we can learn about some really interesting aspects of their biology. So one of the things we've learned is that all of these female-only lizards are entirely formed by mating between different species. So as an example, this is a Eunice species with only female individuals from northern Mexico, and she was actually formed by a mating between a male Arizona whiptail and a female canyon spotted whiptail. So you can see how different these two lizards are just based on these pictures. But if that's not good enough for you, I sometimes like to equate this to what it would be like in primates. Essentially, these two lizards are so different from each other. They are as different from each other as a human is from a gibbon. So this species is essentially the equivalent of a human and a gibbon having a hybrid offspring that can then clone itself a bunch of times. Which is crazy, right? That sounds like a science fiction novel, but it is real biology, real lizards that occur here in the southwestern US where I live. Um, if that's not wacky enough for you, there's also an example of the Chihuahuan spotted whiptail lizard, which essentially is the equivalent of this human gibbon offspring back crossing and mating with a gorilla and then producing a hybrid offspring that can clone itself. So these are what AI art generators seem to think these hybrid animals would look like if they were if they existed, right? So this Chihuahuan spotted whiptail lizard that we have in the southwestern US essentially has three different sets of chromosomes, a set of chromosomes from a human, a set of chromosomes from the equivalent of a gibbon, and the equivalent of a gorilla. So I just find that amazing that in a single individual that that exists, right? Um, so that's a little bit about how new genomic technologies are helping us resolve relationships in the reptile family tree of life and learn a lot about the remarkable diversity and incredible lifestyles of animals that live on this planet with us. So I'll finish by just mentioning briefly how technological advances have changed my teaching. So, like I mentioned, I'm a professor at Arizona State University. I find myself to be very lucky because I get to teach a herpetology class at my university. And one of the things we do is we get to go out in the field and I teach students all about the native species of reptile and amphibians that are here in Arizona. So here are me and a bunch of my students from field trips over the past several years. We've been able to visit some remarkable places and see some really neat species, including even a Gila monster. This is the first Gila monster I ever found in my life in the wild. And so it's a really exciting opportunity for students to be able to see these unique species of reptile and amphibian that live in Arizona. But when we're not out in the field, my students are learning about how we can use freely available online biodiversity databases to actually do their own research and learn about reptiles and amphibians. So in my class, I teach students all about GenBank, this national repository where all scientists who sequence the DNA of reptiles and amphibians deposit their data, which can then be downloaded by anybody to study the genomes of the reptiles and amphibians that they're interested in. We learn about the Global Biodiversity Information Facility, which is a repository that aggregates information about all the records of any species of reptile and amphibian that scientists have collected and deposited in a natural history museum. So students can learn about where species exist on different parts of the earth and generate their own maps. Um, we learned about the reptile database, a really neat resource for learning about all the species of reptile amphibian that exist on Earth. We learn about Morphosource. So this is a really neat, relatively recent online platform where students can download CT scan data, so basically digital X-rays of specimens, and then they can explore the anatomy of any species of reptile and amphibian for which data exists. So here's some pictures of a skull of a caecilian or a really neat cordylid lizard from Africa. You can see that has these really big scales. And so students can begin exploring the morphology of species. We learn about the freely available environmental data, about environmental conditions virtually in any part of the world that students can then download and use to build models and ask questions about how they expect different species of reptiles and amphibians to be active, and at what times of day and at what times of year, using really open source and free statistical software. And there's all a bunch of online, really interesting online learning modules that I have my students learn and do to learn about different concepts that we cover in my class. Yeah, those are three different ways in which um, digital technologies have really changed how I go about doing my research. With that, I should say thank you to you all for having me, and thank you to, I should mention, all the biologists and students who have helped me in the field during my time as a researcher, including this guy right here. This is my nephew, Mikey. He is an excellent lizard catcher and has helped me out in the field a number of times. So thank you all. If there are any questions about anything that, if anything didn't make sense, I'm happy to chat a little more about that. So yeah, thank you all for having me.